हाँ, chat is open है ना? Chat is open ना? Check. I think no one is joined till now. Check it. हाँ, slowly now coming. So, good morning, everyone. Slowly people are joining. Meanwhile, some questions are there. One of the student wrote, "Sir, is there any possibility to reconduct the exam?" So, what are the latest verdict of the Supreme Court? Based on that, they not stop the counseling, right? So, it is very doubtful. Very rare chances is there to reconducting it, right? Very good morning, good morning, good morning to everyone. I think slowly people are coming. So just, sir, offline class strength, I think 6 batches is running. So if you just admitted yourself for offline batches, it is better to come in the campus. So offline strength has crossed more than 1,000. I think 1,1300 it's reaching. Slowly students are coming. Yeah, it is enough. One student is asking, one old gym is enough. It is enough. And this year we in biology and even in another subject, in biology we made the entire new modules. So today topic, see, today we are just going to discuss about some of the basic terminology. You are just facing some of the problems sometime to make a clarification about these terminology. That lecture is going to become very useful. As I told, tracks it we will start from Monday. Meanwhile, these 2-3 days, we are just talking about some basic thing, but very important thing for your understanding of the biology. Right? Is it clear? One student asking, sir, you came from Poranki campus? Yeah, I went there. Till now in boys, 1 batch is almost completed. In girls, I think more than 4, I think 5 is running. Okay, let's start now. So I'm just going to talk about some of the very basic terminology, right? One term I am writing chromatin, chromatid, chromosome, sister chromatid, homologous chromosome, gene. So we will somehow start with these particular terminology. Either you go for cell structure, cell cycle, genetics. In these area, you just find like these terminology are continuously going to used. So we will discuss about these terminology, right? Once, okay, wait. Meanwhile, some questions are coming from your side. So we are starting with these things today. So one questions came from your side. Recent for grace mark given to students. Sir, yeah, I told you in the study class itself. There are so many loopholes was there in this year NEET exam. So many loopholes, right? Not one loopholes. One loopholes is grace marking. Another loophole is conduction of paper in one center in two different seating. Like the Hindi medium student got English paper and vice versa. So they conducted two-time exam. Somewhere it is in Patna, people are saying there was some proof of paper leak also. So many malfunctioning occur this year in the conducting of the NEET exam. Let's see. It is on the court what they are deciding. The things are in court now, but maximum chances is their reconductions not occur, because till now Supreme Court not put any stay on the counseling, right? So you don't waste your time in all these things. What will happen, what not happen, whatever will happen, definitely you have to prepare. Getting the things or not? So don't waste your time on these things. 99.9% chances is there nothing is going to happen. The counseling will start, right? It is unfortunate, but definitely a lot of criticism occur this year. So probably they will take care of these things in the coming years, right? Botany is very difficult to me, sir. It is not difficult. Trust me, it is easy subject. It is easy subject. Right? Diagram questions. See, what I told this year, diagram questions earlier, like 5-6 year, diagram questions was not coming. So from every people side, there was a casual approach on the diagram, right? So don't worry, we will take care of all these things. So just we are going to work about it. So chromatin we are starting with. Are you aware about, if anybody going to ask you a question, what is chromatin? Let's start now. If anybody going to ask you a question, what is chromatin? Then how you will answer it? How will answer it? What is chromatin? See the things. This is a cell. Inside the cell, this is the nucleus. I'm just talking about some basic terminology. First, this is nucleus. Okay? And inside the nucleus, you will find this is an indistinct network is there. And this indistinct network which is present inside the nucleus is called chromatin. So it is a which kind of nucleus? Interphasic nucleus. See, this is basic terminology, but it is important to understand that. The reason I am dealing in these days, interphasic nucleus. So what we are finding in interphasic nucleus means this cell is in which stage of the cell cycle? Interphase stage of the cell cycle. So I am just writing here indistinct network of nucleoprotein present in interphasic nucleus is called chromatin. So till now we just managed to know about कि chromatin is a indistinct network. Indistinct means not well differentiable, not very clearly visible. Got the idea? Not very clearly visible network. And it is forming a network like a structure. See, it is looking like a network. It is like many things, like it is just going to form such kind of a structure. So it is forming a network also. So it is indistinct network, but this network is made up of what? This network is made up of nucleoprotein. So here it is written about chemical composition also. What is the meaning of these nucleoprotein? Nucleoprotein made up of two components. One is what? Nucleic acid. Now another is what? Protein. So in nucleic acid, what type of nucleic acid present here? So here it is a DNA and another is RNA. In protein, how many different proteins are present? So you can say about histone protein. You can say about non-histone protein. You can say about non-histone protein. Getting the idea? So now there is a good MCQ you can make here. Ki what are the chemical compositions of the chromatin? What are the chemical composition of the chromatin, or which of the following is present in chromatin? People can ask a question. So in chromatin, even your NCERT wrote, who is present? DNA is present, RNA is present, histone protein is present, non-histone protein is present. So these all together are present in a chromatin. You got the idea? Is it clear in the mind? So you got the chromatin present in interphasic nucleus, first thing. It is an indistinct network, not clearly visible network. And the chemical compositions of the chromatin is what? Basically, it is made up of DNA, RNA, histone protein and non-histone protein. I hope you got a basic idea about this particular terminology. Chromatin. Is it all right? Is it clear? Is it done? Understood? Can we move ahead on the next? Just I am waiting for your response. So what is chromatin? Chromatin is an indistinct network of nucleoprotein. One student saying, sir, explain again, don't worry. So chromatin is what? If you see a nucleus at interphase stage, inside the nucleus you are finding this is a network like a structure present. When you stain it with the basic dye, you will find there is a network like a structure present in the interphase stage of the nucleus, and this structure is called chromatin. And that's the reason I wrote indistinct network of nucleoprotein present in interphasic nucleus is called chromatin. Got the idea? Then I wrote what is nucleoprotein. So I told the compositions of the nucleoprotein. Nucleoprotein is made up of nucleic acid plus protein. Nucleic acid is what? DNA and RNA. Which type of protein is there? Histone protein is there. So one good question people can ask from here. Chromatin are chemically made up of option 1, or you can say one DNA, another RNA, another histone, another non-histone protein. People can give only 1 and 2, 1, 2 and 3, 1, 2, 3 and 4. All are there. So got the idea? Such type of question people can frame. Got the idea? Now I am asking another question. I am changing the slide. I am asking another question. Just see the board very carefully what I am drawing. Suppose that this is a big nucleus I made. And see the movement of my hand while I am writing. It is important to see. See, I am making one chromatin here like this. It is A diagram, it is B diagram. Just see here. See, as I am starting, I am continuously making a network like this. Got the idea? Here what I am doing, see what I am doing here. I am starting and I am also making a network here like this. This is also a network, this is also a network. But what the difference in the two network? This network is made up of a single bigger thread folded on each other. This network is made up of so many different threads tangled with each other. Got the idea? So in our nucleus, suppose that I am talking about human nucleus and interphase stage. Suppose that I am talking about human nucleus and interphase stage. It is like A or it is like B? Just first give your input. A means, see, once again I am showing you. A means network going to make like this. It is a network. B means so it is A-like or it is B-like. Means there is a single long chromatin are folded on each other and making a network, and so many threads are tangled together and making a network. So in our cell or in a, got the idea, the situation is like this B. This network is, this is chromatin network, and in human cell I am asking another question. In this network many chromatins are there, not a single chromatins are there, many chromatins are there. Now I am asking another question. Now I am asking another question. How many chromatins are present in human somatic cell? And options I am giving: first option 1, second option 2, third option 46, and fourth option variable from person to person. What the answer will you give with this question? What answer you want to give? One, two, three, or four? First option is 1, second option is 2, third option is 46, fourth option is variable from person to person. So some of you are answering 4, some in the answer means variable from, many of you are in favor of variable from person to person. Some of you answer about 2, means 2 chromatins are there. Got the idea? I am telling you the correct answer, but before I am asking another question. Then I will give the answer of both the question together. Then I will give the answer of both the question together. How many chromosomes are present in human somatic cell? And once again, options are same. First option is 1, second option is 2, third option is 46, and fourth option is variable. I am simply writing variable. Now, almost everyone are giving the correct answer. Almost everyone. See, these are all basics. It is important to know the subject. These are all very basic things we are going to deal. Got the idea? So here answer is coming from every of you. Each 46. And it is a correct answer also. Answer is coming 46 and it is a correct answer also. But why you get confused in the upper question? Why you get confused in the upper question? In upper question, I asked how many chromatins. I am asking here chromatin and here I am asking chromosome. In case of chromosome, you people answered it correctly, 46. Well done. But here, I think very few members give the correct answer. Very few members give the correct answer. Here the also correct answer is 46. Now you will ask, sir, how chromatin and chromosome are different and what are the relation between these two? You can ask a question. Huh? Answer of both the question. Either I am asking chromatin or I am asking chromosome. Answer of both the question is 46 only. Okay? Is it all right? Now I am just telling you about why I am writing like this and how chromatin and chromosome are related together. Got the idea? Then you got a better understanding. Isn't it? And it is important to deal. See, right now we have time. So if you have time, you can dissect the things. You can go for a lot of detailings about the things. Getting the idea or not? Otherwise, certain time when tracks it started, certain time we run lack of time. So it is the best time to just make clarifications of these things. Got the idea? Is it clear? Now give your attention. I am just showing you something. See, suppose that this is a chromatin. I draw like this and I am telling what is this? Chromatin. Tin. It is a chromatin. Is it clear? For your information, I am telling, because I will tell later on about it in packaging, but I am giving you one information. Each chromatin contain one DNA. Each chromatin have one DNA. I am not going to amplify right now this structure. I will amplify it when I will teach in the main track sheet. But meanwhile, you keep in mind, you make it as a rule, each chromatin have one DNA. Got the idea? So in this chromatin, who is there? It is chromatin 4 component I told, na? I told you 4 component in chromatin. One is DNA, another is RNA, another is histone protein, another is non-histone protein. So in this chromatin, how many DNA is there? One DNA. In every chromatin, how many DNA is there? In every chromatin, how many DNA is there? One DNA. Don't forget this line. It is an important line. It is important basic things. You must know. Got the idea? Is it clear? All right. Now have a look. Now give your attention. Now what happened? This condition is present in which stage of the cell cycle? Interphase. Chromatin looking like this. But when it will undergoes in division phase, an example prophase. It is going in which phase of the cell cycle? Assume prophase of the cell cycle. So during prophase, what happened? These chromatin condense. Condensation occur. condensation occur in the chromatin in prophase stage. In prophase stage, what happen in the chromatin? Condensation occur in the chromatin. It undergoes in condensation. And our outcome of the condensation is result shortening and thickening. These chromatin become short and thick, and it is going to make such kind of a structure I am drawing. It is condensing, and after condensation, it is going to make such kind of a structure. It become like this. Make it somehow more longer. It become like this. So chromatin becomes shorter and thicker and become like this in the division phase, an example in prophase. This type of chromatin you are finding in interphase. Now we not call this structure as chromatin. Due to condensation, it becomes shorter and thicker, and its name now gets changed. Now it is not the chromatin, now it is called chromosome. So when chromatin undergoes in condensation, it is forming the chromosome. Got the idea? So what is the difference between chromatin and chromosome? You got the idea? Condensed form of the chromatin is chromosome. So I wrote this terminology, chromosome also. So how it is differing from each other, you got the idea? When it undergoes in chromatin undergoes in condensation, it will form chromosome. And when chromosome undergoes in decondensation, it is forming chromatin. You got the idea? You got the difference between chromatin and chromosome or not? Is it all clear? Things is coming in mind? Now, if I ask a question here, got the idea? I will answer these things of your question when you come here, because right now what problem occurred? If I start answering any individual question of a student, you try to understand the technical problem right now. Rest will not able to connect. Ki what questions asked by X student and how I am handling it. Remaining with it is going to got a broken information. So please have patience. So what I am telling, listen it. It is going for condensation and it is going to become shorter and thicker, and this structure is called chromosome. This structure is called chromosome. Is it clear? Is it all right? Done? So if your cell have 46 chromosome, it is only possible for 46 chromosome. How many chromatin require? How many chromatin require? 46 chromatin require, because every chromatin converted into a chromosome. So if 46 chromosome are there, it is only possible when this network is made up of how many? I'm just coming on that diagram. How many threads are there? 46 threads are there. It is like this. You just must be very conceptually rich. So it is one thread, another thread, another thread, another thread, another thread, another thread, another thread. Likewise, how many threads are there? 46 threads are there, and it is making a network like a structure. And each thread have one DNA. Each thread of the chromatin have how many DNA? One DNA. So 46 chromatin thread have how many DNA? 46 DNA. Is it clear? Is it all right? Up to here, can I expect? Are you comfortable enough? So each chromatin converted into a chromosome. So 46 chromosome means 46 chromatin. Done? Clear? Now, I am also talking about some more basics. See, look here. Haji. So, see, this is a long-term batch, and everybody have studied last year somehow the NCERT. In NCERT, one line is written, the length of the human DNA is how much is it written? Answer it very good. Someone giving a smart answer. It is 2.2. They have written. I am adding meter here. It is 2.2 meters. Very good. Now I am asking the question. This is the length of one DNA of the human cell, or it is the length of 23 DNA of the human cell, or it is the length of all 46 DNA of the human cell. 2.2 meter is the length of how many DNA? 1 DNA, 2 DNA, 23 DNA, 46 DNA. I'm making a question from here only. 2.2 meter is the length of how many DNA present in human cell. I am giving a option. First 1 DNA, second option 2 DNA, third option 3 DNA, fourth option 46 DNA. So many answer in coming in favor of sir 1 DNA. Many answer came in favor of 1 DNA. Few students are telling no sir, it is not 1 DNA, it is 46 DNA. Got the idea? Now we are going to decode it. See, give your attention here. The correct answer is fourth here, 46 DNA. Actually, what's happened? Suppose that your 1 DNA is this, another DNA is this, another DNA is this, another DNA is this, and dot, 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 so many, it is 46 DNA. Got the idea? So when we place each DNA side by side, after 1 DNA, suppose that it is another DNA, in the same line, near to it I am putting another DNA, another DNA, another DNA, so it becomes 46 DNA. And when we are measuring it, it is 2.2 meter. In second year NCERT wrote 2.2 meter, in first year NCERT wrote what? Approximately 2 meter. Are you getting the idea? So that conceptual error is over, I hope. Many of you are writing about it is the length of 1 DNA. It is not the length of 1 DNA. It is the length of how much? 46 DNA. Clear? Now I am coming on my previous question. Try to connect with me. Definitely you got a lot of good information. Patiently listen it. I will start the track sheet on Monday. I already told that we are going to start with morphology. But these are the time we have to talk about some introductory classes. So in introductory classes, what to do? Teacher don't know much singing, not much dancing. So we know the teaching only. So we are just talking about some small thing in the introductory topics. Is it all right? So see the things, give your attention, have a look here. Now I am going to talk on what previously I told. I am coming on this diagram. I hope you are able to understand this is one chromatin. And this is what? One chromosome. So condensed form of the chromatin is called chromosome. It is one chromosome. But many of you will ask a question from me. Ki, sir, since childhood I know chromosome somehow differently. What picture of chromosome is there in mind is like this, sir. Many times students say me, sir, you are telling this is chromosome, but I know chromosome look like this. Right? So either this is correct or this is correct. To whom we consider a chromosome? In everywhere, when I see the diagram of chromosome, it is coming like this type of diagram. But you are telling that a chromatin is condensing and it is going to form a chromosome. Then what is this? I hope some of you are thinking in such a way. Is it or not? Such type of questions coming in your mind or not coming? One student asking why prophase of mitosis follow S phase and G2 phase. Because before division preparation occur na. So in S phase, G2 phase, what happened? Preparation of division occur. That's the reason prophase occur after that. I'm just answering one individual questions of a student. They just pin it. I just answered him, getting the things. But I'm focusing on the agenda, what we are talking about right now. It is a chromatin. It is a chromosome. But you may think like, many of you are thinking like, I know chromosome is like this. Then what is this? Now, let me tell you what happened. What are the differences between the two? So here, if I wrote, it is how many chromosome is there? I am asking a question, small question. Wait a minute. I am writing its name is diagram A. I am writing the name of this diagram A. I am writing the name of this diagram B. So in diagram A, how many chromosome are there? So you can say, sir, one chromosome. What big is there? One chromatin was there. One chromatin condensed. It becoming one chromosome. In A, one chromosome is there. It is one chromosome. Here, somehow constricted region is called centromere. And these both sides, it is what basically arms. Here you see one arm is smaller looking, another is bigger looking. It is called which arm? P arm. Shorter arm is called P arm. The longer arm is called Q arm. Now, if I ask a question, in this A chromosome, how many centromere is there? Answer it. In this chromosome, number of centromere, A number of centromere, how many centromere is there? You please tell me. It is one centromere is there. This is the centromere. These constricted area is the centromere. How many centromere is there? One centromere is there. How many DNA is there? One DNA is there. Agree? One centromere, one DNA is there. Got the idea? So the chromosome who consists of only one, and this structure is also called other name. It is called chromosome as well as we call it chromatin. So this chromosome have how many chromatid? Only one chromatid. And this condition of chromosome, for that we use a terminology monad. So this chromosome is called monad. Monad are such chromosome which consist of one chromatid. So I am writing here, what is this monad? Monad are what basically? Chromosome consist of one chromatid. Is called monad. Now I am coming on your main question. I am able to understand. Definitely you are not here, you are not speaking, but we can understand what are in your mind, what you want to ask here. You just want to ask here, just explain this, sir. If it is chromosome, this is what? So I am just trying to give this one as a structure like this. So I am also going to make such kind of condensed form like this. Now it is somehow looking like. So what you are finding? Here, 2 copy of such a structure is there. And these 2 copy of such particular structure, it is copy number 1. Don't confuse, it is copy number 1. I am changing the color. It is copy number 1 and it is copy number 2. Both are joined together at the centromere. Here centromere is only 1. So 2 chromatid joined together at the centromere here. And these chromatid are called sister chromatid. These are called sister chromatid. If you talk in the language of cell cycle, if you see a cell in G1 stage, it have only 1 copy of such a structure. Only 1 copy. But when cell undergoes in S phase, in S phase a DNA replication occur and it become like this. Wait a minute. I am changing the color so you can differentiate it better. It become like this. And what you are finding, this structure is somehow this is the structure made like this. So this is also a chromosome. If anybody asking you, it is in G1 stage, it is after S phase, this is also a chromosome. And this is also a chromosome. This is equal to this. Or I can make like this in a more easier way, you can understand. Just a minute. I can make it like this so you can understand it better. In G1 stage, story is like this. Assume, I am telling assume, after S phase it become like this. Here chromosome duplication occur. Chromosome duplication occur. It's 1 duplicate copy also formed in S phase. But these duplicate copy not get separated. They still remain attached at the centromere. Got the idea? So this chromosome, it is also 1 chromosome, it is also 1 chromosome. This chromosome is called monad and this chromosome is what basically dyad. What is monad? Chromosome with 1 chromatid. What is dyad? Chromosome with 2 chromatid. So here what happened? Duplication occur. Duplication of chromosome. But beside of chromosome get duplicated, number not increase. Here how many chromosome is there? 1. Here how many chromosome is there? 1. We can't say 2 because it is not separated. We can't say 2 because it is not separated. Are you getting the idea? Understanding the idea? Clarity coming in mind or not? It is 1 chromosome. It is 1 chromosome. It is not 2 chromosome. Is it clear or not? Is it done? So number of, I am giving you a tricks. It will help you to identify it. Number of chromosome always equal to number of centromere. So in this cell, how many centromere is there? How many yellow dot are there? I am just telling here, I am just telling it is one centromere is there. Here also how many centromere is there? One centromere is there. So number of chromosome always equal to number of centromere. Here how many chromosome is there? One chromosome. Here also how many chromosome is there? One chromosome. Here how many chromatid is there? One. Here two. Here how many DNA? One. Here two. So what happening? In S phase, number of chromatid becoming double. In S phase, amount of DNA becoming double. In S phase, number of DNA becoming double. But in S phase, number of chromosome not changing, it remains same. See, one chromosome, one chromosome. One chromatid, two chromatid. Here it is one chromatid, don't confuse. I am just putting a line. It is one chromatid. Here how many chromatid? Two chromatid. One this, another this. So one chromatid, two chromatid. And these chromatid who attach with the centromere is called sister chromatid. I will tell you later what is sister chromatid. You got the idea? So monad stage or dyad stage, you got the differences? Many time in very good interviews, interviewer asks the candidates, how many chromosomes are present in, how many DNA is present in a chromosome? In many interviews, such question going to ask. Even in interview, I am telling, Viva, like UPSC, like for exam who are just going to make a professors in the colleges, in the government colleges, even like the university colleges. People ask, how many DNA are present in a chromosome? So, answer depend upon situation. If you have to opt out of many options, you have to opt how many? One. One chromosome have one DNA. But if a answer is coming like it variable depend upon stage of the cell cycle. See, in G1 stage, a chromosome have, in G1 stage, a chromosome have how many DNA? One DNA. In S phase, a chromosome have how many DNA? In S phase, a chromosome have how many DNA? Answer it. 2 DNA. In G1 phase 1, in S phase 2. So it depends upon stages of the cell cycle. But there is a thumb rule. One chromatin always have one DNA. There is a thumb rule. You don't forget it. 2 line I am writing and you please note it down. One chromatid always have one DNA. In one chromatin, one DNA is present. Got the idea? Number of DNA in a chromosome? In a chromosome. So if it is monad, 1, if chromosome look like this. If it is a dyad, if chromosome look like this, 2 DNA. You got the idea? These understanding came? These understanding came? If chromosome look like this, it is 1 DNA. If chromosome look like this, here it is how many DNA? 2 DNA. In dyad stage, 2 DNA is there. In monad stage, 1 DNA is there. Clear? So this area is over. Done? Now we are moving ahead. So you got the monad, you got the dyad, you got the chromatin, you got the chromosome. This terminology is over. Right? Now I am asking a question. See, this is one chromatid. I am changing the color. So this is another chromatid. This is another chromatid. This is another chromatid, but one thing is they are all joined to a common centromere. How many chromosomes is there? Answer it. All joined to a common centromere. How many chromosomes is this? Tell me the number of chromosomes. Number of chromosome will be one only, because I told a rule to you. What? Number of chromosome always equal to number of centromere. So how many chromatid is there doesn't matter. All are connected together, na? All are connected together, na? Getting the things or not? So as long as both are connected together, you cannot say about it is more than one. So number of chromosome always equal to number of centromere. So now I hope you will not make a mistake from this area. If any glitch is there, here DNA is how many? Here how many DNA is there? This is a abnormal condition. It is not a normal condition. Here how many DNA is there? In this one especially, how many DNA is there? If anybody going to show you a diagram, how many DNA is there? 1, 2, 3, 4. In this question only, not in all question. Done? Over? Now we are moving ahead. Give your attention. Now I am coming on the terminology. What chronology we set here? Let me check. So chromatin, can I put a tick mark? Chromatid, can I put a tick mark, or you are still confused about chromatid? See, it is chromatin. Condensed form, it is chromatid or chromosome. You can say chromatid or chromosome. Getting the things? And chromosome, you also know about it. You know about centromere, you know about telomere. I am just asking another question. Wait. It will also help you when you will read the respective topic. Let me make some good diagram. Wait a minute. Deliberately I am changing the color, so you can differentiate between the two chromatid. One color is one chromatid, another color is another chromatid. This is a chromosome. This is a chromosome. It is, I am asking question, you answer it. It is monad or dyad? First answer is, it is monad or dyad? Hurry up. Very good, it is a dyad. What is the number of chromosome? In this, suppose that it is diagram number A. What is the number of chromosome in this diagram? Let me ask the final question from here. Number of chromosome. Number of chromosome is 1. Well done. Now I am asking number of chromatid. Number of chromatid. Number of chromatid. Good one. Answer is coming 2. One is yellow color, another is blue color. For your understanding, I made 2 different color here. So how many number of chromatid? Number of chromatid is 2. Done? Over. Number of chromatid is 2. Now I am asking question, number of DNA here. Number of DNA in this chromosome. Number of DNA in this chromosome. 2. Because I told 1 DNA at the rate of each chromatid, always. One DNA at the rate of each chromatid, always. So how many chromatid is there? 2 chromatid is there. How many DNA will there then? 2 DNA. Done. Over. Next question I am asking, number of centromere. Number of centromere. Number of centromere. It is not 2. Centromere is this connected area where both are joined together. How many number of centromere is there? Here it is 1. That is the reason number of chromosome is 1. Number of centromere 1. And terminal end of a chromosome is called telomere. For your information, I am telling. Here, this is 1. This is telomere, this is telomere, this is telomere, this is telomere. So I am asking a question, number of telomere. Tell me, number of telomere, how many telomere is there? Every end is called telomere. So how many end you are finding? 1, 2, 3, 4. Getting the thing? So number of telomere is how many? 4. Now, I am just giving you a basic structure about the chromosome, such type of question. In chromosome, here a proteinaceous disc shape structure is present, like this. And this is called kinetochore. So in dyad stage here, how many kinetochore are there? Number of kinetochore. So some numerical type of question over na? I hope you are understanding. So how many number of kinetochores are there? Tell me. Number of kinetochore, this proteinaceous disc shape structure is how many? 2. So what is centromere? If someone asks you a question, if anybody going to ask you a question, what is centromere? What answer will you give? And even anybody going to ask you, what is kinetochore? What the answer will you give? Such type of things come, na? And we not take care while teaching. We just simply mugging the things, moving the things. Understanding is important. If you develop the understanding, things become very easy in your life. So first tell me, what is centromere? I hope first of all, every student, I am asking to everyone who are attending the class, are you really comfortable here? What I wrote these values, are you really comfortable here? Are you comfortable? Yes, well done. Right. Now I am coming on this question. What is centromere? So it is a constriction present in chromosome. And it is also called primary constriction. Centromere have another name also, primary constriction. Present in a chromosome. 2 sister chromatid join together at centromere. So whenever I am making a chromosome in diagram, Just look here. I am making a chromosome here. I am making a monad only. You can see everywhere it is not of the same thickness. Here thickness is less. So this is a constriction, and this constriction is what basically centromere. And we called also it as what? Primary constriction. So keep one thing in mind, centromere is a constriction, it is also called primary constriction present in a chromosome, and at this constriction two sister chromatid join. These yellow and blue color structure I made is a sister chromatid, they join together at this place, at this constriction. Got the idea? So centromere clear? Is it all right? Now kinetochore. What is this kinetochore? Kinetochore is a proteinaceous disc-shaped structure. Proteinaceous disc-shaped structure, and at this proteinaceous disc-shaped structure, who is going and coming to join? A spindle fiber came and join. So spindle fiber join at kinetochore. Two sister chromatid join at centromere. Don't forget, this is important information, easy but important information. Spindle fiber join at kinetochore and two sister chromatid. It is easy but important information. Join together at centromere. Is it clear? Is it all right? Is it done? Now I am asking another question. Can you tell me any differences between this yellow chromatid and blue chromatid? Both are sister chromatid. These yellow and blue are which kind of chromatid? Sister chromatid. What are the differences between the two? I am asking a small question. Give your input. Just last question. Time over now. And now very honest question I am asking. Is this session was useful or you want somehow differently in these days as long as track sheet is not started? Color difference? What you are answering, dear? Color I made only what? To differentiate between it is a chromatid, it is another chromatid. I make a color for which purpose? To differentiate it is a chromatid. Otherwise, many times students confuse. How they confuse? If I draw a diagram, many times some student think, yeah, this is a chromatid. Mean that part only. Many times student confuse, this is a chromatid. So to rectify that confusion, I draw two different color. Ki bhai, yellow color one chromatid, another. So you can say like this is one chromatid and this is what? Another. I use the color to say you which part is chromatid. Getting the things or not? Isn't it? Any differences are there between these two sister chromatid? Answer is no. Sister chromatids are identical. What information present in this chromatid? Exactly same information present in the another chromatid. Because both have same 100% same DNA. Formation of one sister chromatid occur by replication of DNA. And when one DNA replicate, two DNA is forming and both are 100% identical of each other. Got the idea? So there are no differences. Sister chromatids are identical. Keep in your mind. They are 100% same. No any differences are there in between these two. Clarity came in the mind. Understanding came in the mind. Okay, ji. Now, another question I am asking, who are available from home, these session are such kind of session is useful these days, or you want should we do some different thing like paper discussion of this NEET, because we start the track sheet from Monday. So if you are finding no sir, it is a useful session, I will go for such kind of sessions. Getting the things or not? Otherwise, we will discuss about the 2000 NEET paper. Suppose that many of you have not appeared in the NEET, so, or it may be you want to discuss the paper explanation, I will go for paper explanation. So what it is there, we have how many days? Thursday, Friday, Saturday, 2 days is there. So one day I will go for paper explanation, another day we will go for such kind of things, and then of course Monday onward our track sheet will start. And it will start from which one? Morphology. So I expect from you before starting, you just start with a bang. Start with reading the NCERT morphology. See, you are writing the exam this time, second time. So it's not like any new thing you will find. You know many of the things, but the things require you must know with the perfection. My pet dialogue is there. What factual information is there, it must be in the language of 2 to 4. If this perfection is missing, it is not good. You will confuse and you will do the mistakes. So in factual area, these perfections require. Got the idea? Thank you very much for your patience for listening. Bye-bye. Have a good day to all of you. Thank you, thank you, thank you. In offline mode, also main track sheet start from Monday. Okay? So whoever planning to come, better you come in the campus faster. Thank you very much.